college level. God is calling you to band. For God, everyone's for which we stand. Red for a promise in the game. White for the glory of a heart that's pure. Bright with an ever shining flame. Alma Mater, we hail. God give the earth and great success. With truth and love will conquer all. For God a land and SFS. Alma Mater, we hail. God give the earth and great success. With truth and love will conquer all. For God a land and SFS. With truth and love, let us unite in peace. To our glorious college meet. High on the hill, a beacon light is sheet, leading our children on to faith. March on to glory is a message for spread truth and love will far and wide. Loyal to the hands who reigns of guidance, teacher and pupil side by side. Alma Mater, we hail. God give the earth and great success. With truth and love will conquer all. For God our land and SFS. Alma Mater, we hail. God give the earth and great success. With truth and love we conquer all. For God our land and SFS.
Hello and good morning to all. This is Dr. Susan John, Assistant Professor, Department of Physics, SFS College. We, the SFS management and faculty, are indeed happy to host this webinar on recent developments in genetics and their impact on religion and society. The sequence of today's program is as follows. Today we have enlightening talk by our distinguished resource persons, Dr. Firoz Hussain, Dr. T. Srinivasu, Dr. Victor Ferro, followed by concluding remarks by Father Job K. Now we are moving towards our first session. We are happy to have with us the resource person of the first session, Dr. Firoz Hussain, Principal Scientist, Maize Breeding, Division of Genetics, Indian Council of Agricultural Research, Indian Agricultural Research Institute, New Delhi. Let me briefly introduce him. Dr. Firoz Hussain is serving as the Principal Scientist in the Division of Genetics, IARI, New Delhi. He is currently the Program Leader of the Maize Breeding at IARI. He is pursuing his research in the field of nutritional quality enhancement and specialty traits. He and his team have developed 7 biofortified maize hybrids using molecular breeding. Of this, Pusa Vivek QPM9 Improved has the distinction of being country's first pro-vitamin A rich maize hybrid. He is also a part of 3 multi-nutrient rich maize, lysine, tryptophan and pro-vitamin A rich hybrids released for various agroecologies of the country. He has also developed maize hybrids with improved vitamin E and higher bioavailability of iron and zinc. Dr. Firoz has developed 2 sweet corn hybrids, which is Pusa Super Sweet Corn 1 and Pusa Super Sweet Corn 2. He has recently developed two biofortified sweet corn hybrids, again first of its kind in the country. These biofortified maize hybrids serve as the vehicle to alleviate malnutrition. We are indeed privileged to have such a distinguished scientist with us today. A warm welcome to you, sir. Now I kindly request Dr. Firoz Hussain to take over the session. Over to you, sir. Firoz, sir.
Can you see the slide?
Yes, sir. Yes, sir.
Okay, a very good morning to you all. At first, I'd like to thank SFS College Nagpur and their authorities for giving me this opportunity to give a overall development of nutritional maize as far as the breeding is concerned. I'd also like to thank Dr. Susan John, who contacted me and then to be in touch for in several times so that this information slides and all these are in proper order. So I thank you all. So I'm basically presenting the work on nutritional quality enrichment in maize through genetic avenues. Now, we all know that malnutrition is now a global problem, and around 2 billion people, we have around 7.5 billion people, so around 2 billion people are malnourished. Several millions of people are undernourished, and children are basically less than 5 years. They have a serious problem of stunting, wasting, and in fact severe wasting, which leads to almost loss of 11% of GDP in Asia and Africa. And if you look at the figures of children being affected for stunting, overweight, or wasting, which are the indicators of malnutrition. We normally see that possibly Africa being somewhat kind of a poor nations, they will be having major problems. But actually, the problem is more in Asia, because if you can see in stunting, Asia has got 55% of the children below 5 years as having the problem of stunting as compared to 39. And similar is the case with wasting also. So it does mean that we have to really give urgent attention to address this problem of malnutrition. Now, this is the map of global malnutrition. We basically have the major deficiency is basically on protein, iron, zinc, and vitamin A. And of course, we have a problem of other minerals as well, but these are the major problems. And if we look at our country basically in the alarming zone, that is almost in the deep orange zone. Sorry, can you see the cursor on the slide, please? Can you please someone tell me?
Yes, sir. We can see your slide, sir. Hello?
No, cursor. I'm telling the cursor where I want to point out.
No, sir.
Is it visible?
Sir,
the mouse is not visible, right?
Cursor is visible, sir. I saw the arrow. Yes, sir. It is visible.
Oh, okay, then that is fine. Good. So what essentially I want to mention is that India falls under a severe category, and therefore we need urgent attention. And in many places, there is not only one type of problem. There are double burden problems, there are triple burden problems. What do you mean by single burden, double burden? Anemia in adult women and overweight in adult women. These are the three major parameters. So it's not that each country will have either of these three. There are many cases where there are two together. There are also cases there are three together. There are many African nations which are in deep orange. They basically have a triple burden. That means all three problems are there. In India, it is basically two together. That essentially is the anemia among women, and we all know that stunting in childhood. These are the two major problems in India. So therefore, we have a both-sided problem happening together. The United Nations actually came out in 2015 a Sustainable Development Goal, and they had almost 17 goals to actually get rid of poverty, hunger, and malnutrition. But of which, if you can look into, for each goal, there are some parameters, there are some indicators. And orange basically indicates the parameters or the indicators which are related to nutrition. Suppose, for example, goal 2 is to eradicate hunger and nutrition. To hunger and nutrition, there are 7 indicators which are related to nutrition and 7 indicators which are not related to nutrition. Similarly, for healthy lives, we all know to have a healthy life, we need a nutritious diet. So out of these 12 indicators are related to nutrition, while 14 are related to non-nutrition. Similarly, for gender equality, to have our both male and female or girl child or the boy child to be going together with equal emphasis or impetus, we need to give proper nutrition to both of them. We cannot discriminate boy or boy versus girl or something like that. So therefore, nutrition has to be a key issue so that boys and girls will have the equal chance to be to flourish in this whole world. So like that, for each and every goal, there are indicators which are related to nutrition. So if you can see out of 17, 12 goals have orange bars. That means they are related to nutrition, which essentially mean that nutrition is core to alleviate malnutrition and to make our society happy and wealthy and prosperous. If you look at the loss of GDP, India loses around 12 billion US dollar in case of, as far as GDP is concerned, because of malnutrition. It's a huge amount. Our neighboring countries and other countries have also got their losses. But the most important factor is that if we can address this malnutrition, if we can invest $1, the benefit which we get from $1 is equal to $16. That means 1 is to 16. That essentially means that if we can invest in a proven nutrition program, that means suppose we want to give a good food to the children in school through midday meal and other options, the children will have a better chance to grow better and to do well. And the benefit will be equal to $16. So if we can invest $1, we'll earn $16 in returns. It's a huge return. So therefore, nutrition is definitely a key factor. If you look at the malnutrition scenario in India, it is again very, very alarming. If you look at around 22% of the population lives in extreme poverty. If you look at the stunting, around 38.4% of the children below 5 years are stunted, and 21% are wasted. Wasted essentially means the lean. They are not so they're lean as far as their height is concerned. And of course, anemia is a major problem. 58.4% of the children at the range of 6 to 59 months, 53% of the adult women and 22% of the adult men are affected due to anemia. We normally think anemia is a problem in women. It's not the case. Around 23% of the adult men are also affected because of anemia. Iron, 70% of the children below 5 years are affected due to iron deficiency. 38% of the children are affected due to zinc deficiency. So then this is actually the huge problem. And this is what somewhat the maps of India where stunting has been mapped. We have around 604 districts, of which 239 districts have got stunting level above 40%. That is very, very alarming. And this stunting varies from 12.4% to 65, as high as 65%. That means we do not have any district which is not having any problem of stunting below 5 years of children. Normally it is said children, if they are healthy by 5 years of age, they would most likely to have a healthy life in future if they get the proper nutrition. But if they're not having healthy lifestyle before 5 years and they suffer from malnutrition, it will be very, very difficult to come to overcome it. So that kind of problem will always be there. So below 5 years is a critical point. So the whole map suggests that the stunting is a problem almost everywhere, whether we talk about any district. But then there are some districts where there are deep orange colors indicate the stunting is a major problem, even in our country. How to tackle this malnutrition? This is called hidden hunger. That essentially means when we suffer from not hungry per se, if we suffer from micronutrients like vitamins or minerals, that gives a problem. So that is called hidden hunger. Our stomach may be full because of taking rice and wheat, but that food is not nutritious enough, then we'll suffer from a term called hidden hunger. So we may not be per se hungry, our stomach will be full, but then we will be suffering from malnutrition because of the lack of vitamins or minerals. There are various avenues are there. We call it supplementation. We have given, vitamin A pills, calcium pills, and so on and so forth, iron pills. Then we have commercial fortification, like iodized salt is a classical example. Salt is something which you have to take through every food. So therefore, if the iodine is added to salt, then we get iodine rather than taking to other means. There are other method called dietary diversity. That means if we take every kind of food, then our diet becomes balanced. But imagine in a poorest of the poor places, this dietary diversity is a luxury. They don't have or maybe a fortified atta with iron, zinc, folate, or even iodized salt. And many times to receive the pills through iron pills or through vitamin A rich pills is a problem because it requires a lot of effective distribution systems and so on and so forth. But biofortification is a process where a crop variety is inherently improved for these minerals or vitamins. So now we develop a variety which is as good as normal variety, but they are rich in nutrients, maybe vitamins, minerals, proteins, and so on and so forth. And so that is called biofortification. So biofortification is most effective, it is most cost effective, it is most sustainable. And once you develop a variety, once a poor farmer grows that variety, if he can consume that grain, so he will be automatically consuming balanced nutrition. So that is the advantage of biofortification. Now I come to maize. Maize is an important source of livelihood in India and around the world. The world we have around 1100 metric tons of maize is produced, highest among the cereals. Asia produces almost one third of the maize. India produces almost 28 million tons of maize. And if you can look at the usage, it is used as, of course, as a food in some part of the country, but as a processed food in different forms, whether it's a popcorn, nachos, and many forms, we consume consume maize as processed food. But majority of the maize goes to poultry and the livestock feed. So therefore, if you consume eggs for the non-vegetarians, that means if the egg is nutritious, which comes where the chicken gets the nutrition from maize, so indirectly maize is contributing to our nutrition. And now coming to plant breeding, I understand college deals with several disciplines, including physics, chemistry. It may be a little bit difficult for someone who is from physics to understand what basically we do in plant breeding. So just I want to share here. We have a variety which is, say, virus resistant. There is another variety which is high yielding. What we do, we cross them and bring both the traits together so that we have now virus resistant and high yielding varieties. So to bring all the good traits together, the process which we use is called actually plant breeding. Now, once we grow the plant, we select the plant, maybe this is a desirable plant by different means, either through phenotypic features, or now we have started tracking the plants through genes. Now, essentially what is done, so there are these round that suppose genes. This is a desirable gene, which is in blue, and this is a recipient DNA, that means a plant which is not having that desirable gene. Once we make crosses, then this blue desired gene is now transferred here, where we have some green part and orange part. So we get now an improved variety. That is what it is. So plant breeding essentially means that we transfer gene from a donor plant to a recipient plant, simple by crossing. We do not use other method. Normally in transgenic, a gene from bacteria, virus, or any other places can be transferred to plant. Here in plant breeding, we use the natural variant of the gene, which is anyhow otherwise present in the crop. We simply make crosses and transfer that gene. So this is what essentially is the plant breeding. Now, coming to maize, we have a protein maize. What happens? Maize has got around 10% protein, and the protein is basically made up of amino acids called lysine and tryptophan. These are the two essential amino acids. Actually, there are 20 amino acids which makes our protein. So out of 29 amino acids are called essential amino acids because our body cannot synthesize such amino acid inside the body. So we have to take those amino acids through diet. So among these 9 essential amino acids, we have lysine and tryptophan are the two which are deficient in maize. So we have a gene called OPEC2, which basically, if you can introgress, so our lysine and tryptophan level is enhanced. I'll show what happens. We have released a variety called Pusa HM4 improved in 2017, and it has been released for Northwestern Plain Zone, which includes Punjab, Haryana, Delhi, Uttarakhand, Uttar Pradesh, Western region. We have higher tryptophan. Normally, tryptophan level is 0.3. Here it is 0.9. The lysine level is 3.62. Normally, it is around 1.5 to 2%. So here, the lysine and tryptophan has been increased by almost double On the other hand, it remains to have high yield, almost 6.5 tons yield it produces. So that means this particular maize hybrid is having a better nutrition. It provides better protein quality. So if we can consume this maize over traditional maize, our nutritional balance in the diet will be much better, we grow healthier, and we'll be much happier, because health is wealth, we always say. So this is one of the hybrids which we released from Indian Agriculture Research Institute, New Delhi, where I work. There is another variety called Pusa HM8 improved. Here also we have improved lysine and tryptophan, but it was released for other states like Maharashtra, Karnataka, Andhra Pradesh, Telangana, and Tamil Nadu. Actually, in India, we have around five. We have actually 5 zones. Each zone has some states. So when a variety is released, it can be released for across India. It can be released for some of the zones, depending upon its performance. So here it was released for the southern states. We have another hybrid called Pusa HM9 improved. Here also we have better lysine and tryptophan. It has been released for the eastern states: Bihar, Jharkhand, Odisha, Uttar Pradesh of the eastern region, and West Bengal. Here lysine tryptophan level is again much higher, almost twofold as compared to the traditional maize. Now coming to the carotenoids, we know that in maize we have several shades of colors. This is the white maize, this is yellow, this is orange, this is light yellow, dark orange, so on and so forth. Normally what we feel that we must have heard the term carotenoids. Carotenoids is the color or the pigments which gives the orange color. In white maize there is no carotenoids. And it is the carotenoids, basically carotenoids has got several components like lutein, beta-carotene, beta-cryptoxanthin, and zeaxanthin. So they basically give the color. But if you can look into the overall features, it was found that beta-carotene, which gives actually the vitamin A, is only 3%. But then there are also, that means although it is orangish in color, its vitamin A content is very, very low. So there are 2 genes which we call as CRTRB1 gene, which actually increases beta-carotene. There is another gene called LCY, which increases again beta-carotene. There are 2 classical papers came in 2008 and 2010. And we use these 2 genes and developed the country's first vitamin A rich maize hybrid. So here we improved the original traditional hybrid called Vivek Hybrid 27, where it is 1 ppm vitamin A, and our input version is almost 5.5 ppm. So almost 5 times increase of vitamin A is there, and it has been released for again West Bengal, Bihar, Eastern Uttar Pradesh, Odisha, and Jharkhand. We have combined vitamin A, lysine, and tryptophan. So we are not only working on single nutrient, we are also working on multiple nutrients. So this Pusa Vivek UPM9 improved. It has got high vitamin A, almost 8 ppm as compared to 1 ppm in the traditional varieties. It has got double the amount of lysine and tryptophan. So it is much more balanced than traditional maize, and even more balanced than maize, which is having only improved for only 1 nutrition. Here it has been improved for the multiple nutrition. And this happens to be country's first vitamin A rich hybrid, and of course world's first multi-nutrient hybrid where both vitamin A, lysine, and tryptophan has been enhanced. In 2020, we have released another 2 hybrids, Pusa HQPM5 improved and HQPM7 improved, where again the vitamin A, lysine, and tryptophan has been improved through breeding approaches. Now that means these varieties are rich in nutrients which are essential for our body, because our body cannot synthesize vitamin A, our body cannot synthesize amino acids called lysine and tryptophan. Amino acids are nothing but building blocks of protein. So there are 20 different amino acids which makes our body protein. That means we have our muscle. Muscle is made up of our protein. Every day's work, muscle goes through that's years. So it has to be repaired in every hour actually. So for that we need to synthesize protein. So this is very, very important. So we should get the protein through diet, which should be balanced. And this is what is the scenario of provitamin A rich maize in the world. And this orange indicates that the countries have developed vitamin A rich maize. So in India, we have developed 4 hybrid maize. We are the only institute in the country that have come out with vitamin A rich maize. We have released 1 in 2017 and 3 in 2019-20. So our country is now figuring the world to have one of the countries to have released vitamin A rich maize. The maize is grown all over the world, but vitamin A rich maize is not grown everywhere because it is a recent development. India is one of the first nations to come out with this vitamin A rich hybrids. We're also working on vitamin E. This is the pathway. We have transferred the gene called VT4 through breeding. And look at here, alpha tocopherol is an indicator of vitamin E. So this red bar indicates the original hybrid. We have improved that hybrid for this gene, and the alpha tocopherol level is almost double. So we have improved 4 hybrids. So you can see the alpha tocopherol level is increased by double. And these hybrids are rich in vitamin E, it is rich in vitamin A, it is rich in lysine, it is rich in tryptophan. There are 4 nutrients which have been added in these hybrids, and which is much better than the traditional hybrid. Look at the values. We have our traditional hybrid 6 to 8 ppm. Here it is almost 16 ppm. Vitamin A, it is having 1 to 2 ppm. Here it is almost 11 ppm. Lysine, tryptophan again double. So we are the first institute in the country to come out with so many traits, nutritional traits together. So that means this is the most important bio-fortified hybrid. If we want and consume, they'll get much more balanced diet as compared to the traditional maize. They also released sweet corn hybrids. We know if we go to malls and everywhere, then nowadays sweet corn kernels are boiled and they're given with butter and other masalas. It's now become a good snacks. But then this sweet corn is also not having desired level of vitamin A as traditional maize. So we have improved this sweet corn hybrid. We have released a hybrid called SK1. It has been now released for QPM. QPM means lysine and tryptophan. And also it has got high vitamin A. So it has got 5-fold high vitamin A. It has got almost 2-fold high lysine and tryptophan. This is the original hybrid, and these are the 3 different versions of the improved hybrid. So the level is definitely much higher than the original. So we now have sweet corn hybrids which are also rich in nutrition, that is vitamin A, lysine and tryptophan. Even lysine, tryptophan and amino acids which gives better protein quality. Better protein quality means if we can consume that protein through food, then we have a better nutrition. We're also working on iron and zinc, and what really happens is maize has got around 25 to 30 ppm of iron and 20 to 25 ppm of zinc. And what happens? Only 5% of the iron and 20% of the zinc is only bioavailable in the human and animal gut. Bioavailable means only that much portion will be absorbed. This will go through our excreta. So 5% iron absorbed means 95% go as waste through our excreta. Now, the reason is there is a salt called phytic acid, we call it phytate, that basically absorbs the iron and zincs and make them unavailable. Right? So what we do, there are 3 genes, LPA1, low phytic acid, that's why it's called LPA. So there are 3 genes called LPA1, LPA2 and LPA3. We have reduced the phytic acid from 80 to 90% to almost 50 to 60%. And thereby we have increased the bioavailability of iron and zinc. That is what we are doing. And we have several of hybrids which have been released for several zones are now being. Introgression is a breeding term. It means basically to put a gene inside a plant. Introgression means that. So we have now hybrids where low phytic acid mutants are there. Mutants means mutant genes are there. So if low phytic acid mutant genes are there, that means it will reduce the phytic acid. If it reduces the phytic acid, more iron and zinc will be bioavailable. That's what we are doing. So our hybrids are almost ready with high iron and zinc. We have also tested our QPM hybrids on poultry feed, on poultry bird. There is a poultry bird called Vanaraja. It has been released it is a breed which has been developed by one of our ICR institute called Directorate of Poultry Research in Hyderabad. And we have given different diets. Normal means it's a traditional maize. QPM means, of course, it is only QPM. But it shows if our hybrid is rich in both QPM and vitamin A, its body weight will be almost as good as the other one, but it leads less feed conversion ratio. FCR means feed conversion ratio. Lesser feed conversion ratio means it will require less amount of feed to produce similar body weight in terms of meat. So that means this has been proven well. QPM and vitamin A, if we can fed to chicken, so it will be much more profitable business because it will require less feed to grow the similar body weight as compared to the traditional maize. Another very important factor for the non-vegetarians who take chickens, we normally like a chicken, the skins which should be yellow as compared to the white. That is the international, references. So a chicken which is fed with vitamin A rich maize or vitamin A rich maize can develop more pigments as compared to chicken fed with white maize. So therefore there is a preference. On the other hand, if we consume egg, now look at here, there are different maize was fed to the chicken. And this is a white maize. The egg yolk is almost white. There are different shades of yellow. But here there is a PVA. That means it is having a commercial which is having 0.33 ppm accumulation of vitamin A in the egg yolk. And look at here. This is a high carotenoid maize where it is almost 3 ppm. That means 10 times more vitamin A is accumulated in the egg yolk. Where from the vitamin A has come? It has come from the maize, because in poultry, maize is one of the important components of the maize feed, of the poultry feed. So if we can feed the chicken through biofortified maize, there are two advantages. Advantage one, skin color will be better, which is a preference for consumers. Another is the eggs laid by the chicken will have better vitamin A or nutrition as compared to the traditional maize. We have also tested our maize through National Institute of Nutrition at Hyderabad, and we found there is a term called RD. RD means recommended dietary allowance, means how much we require per day. It has been found that our provitamin A rich maize provides around 50 to 64% of the RDA through maize. That means only 40% you have to get from other sources, which is anyhow have to be done. Nobody can depend on only on maize as a food. You have to depend on many foods, but you look at here, 50 to 60% of the vitamin A is met from maize. If you can put it as a part of our diet. And this maize is not a traditional maize; it's a vitamin A rich maize which we have developed through breeding methods. It has got another advantage. Now, what happens if the maize grains are not stored properly? This fungus comes. That fungus produces a toxin called aflatoxin. Aflatoxin is carcinogenic. It creates a whole lot of problem in the health of humans as well as the poultry bird. So what happens is vitamin A rich maize, which we have developed, it has been found to have less aflatoxin infestation as compared to the traditional maize. So that means it has got also additional advantage that it is having lesser contamination of aflatoxin as compared to the traditional maize. And there are a lot of impacts. Now, impact is most important. I mean, a society can only be healthy. The overall topic, what I understand of this webinar is all about health and other aspects of it. So what happens if human consume vitamin A rich maize? So there are several international papers have come out which shows that biofortified maize increases the serum carotenoid concentration in the Zambian children. And again, they have found if vitamin A rich biofortified maize is consumed, the blood has got more retinol. Retinol is nothing but vitamin A. It's a chemical composition of or chemical structure of vitamin A is called retinol. So it has been found that if you consume vitamin A rich maize, vitamin A level increases in our body. Similarly, the impact of QPM. QPM means the maize which has got higher protein quality, that means having high lysine and tryptophan. And it has been found that children who has been fed with quality protein maize or the biofortified maize, they have higher growth, they have better height, and they have lesser fever days. Fever days means more the fever day per year, more is the problem. They have lesser fever days. Lesser fever days indicates they have more immunity, they're better, they're much healthier. So these are all have been proven internationally that if one consumes biofortified maize, the children will grow better, the society will be much better, and possibly that is the most important thing. Here these are my last few slides, very important slides. In 2000, was awarded with World Food Prize. World Food Prize is basically a prize which is regarded as a Nobel Prize in agriculture. So because they have developed this biofortified maize, these two people were they are the maize breeder and maize biochemist. They were given with the World Food Prize in 2000. Then in 2016. Andre, Mangwa, Low, and Buis, 4 people were awarded for their work on improvement of biofortification or the biofortification in crops. But then the policy support is also equally important. So Dr. Adesina, he was one of the policy makers in Nigeria. He was awarded with World Food Prize because he has promoted this biofortified maize. One is the development part. We scientists can only develop. Another is the policy part. The policy part is very important. The government has to promote such kind of biofortified crops or any other crops has to, into a daily routine so that we get better advantage. Similarly, in 2018, Haddad and Nepal also got the World Food Prize for their global leadership in elevating maternal and child nutrition. The most encouraging part is the present government is very, very pro to this biofortification program. We have different rural transformation project by NITI Aayog. We have National Food Security Mission, Rashtriya Krishi Vikas Yojana, Integrated Child Development Scheme, and these are some of the schemes where government is promoting the nutritious food to the children, to women, so that we have better society. In 2017, Indian government came out with a national nutrition strategy, which basically says that to have a nutritional security, we need drinking water and sanitation. We need food. Food means here nutritious food. Income and livelihood should be there, and health services will be there. So therefore, development, awareness means even though it is available, if we are not aware, we are not going to consume it, right? So awareness is very, very important. And then is the policy. The government's policy matters. Policy also matters a lot. So these three components, development, awareness, and the policy, leads to the successful implementation. So this is what was my last slide. I just want to thank my organization, Indian Council of Agriculture Research. I thank Dr. A.K. Singh, Director IRI. In fact, Dr. Suzanne contacted first our director for such lecture, and sir in turn told me to make a presentation. So I thank sir. Thanks to my head under whom I work. I thank IMR, Indian Institute of Maize Research. Then there are various institutes who actually provides the support. There is DBT, DST, CRPR, funding agencies for different projects. CIMIT and USDA, they provide the donor germplasm. I sincerely thank Dr. Vignesh and Rajkumar. They are the two scientists in our program. They are one of the backbones of our program to develop such varieties. We thank all our collaborators, students, and research fellows. And of course, at last, I once again thank Dr. Suzanne for giving me this opportunity to give a little bit of glimpse of how we work in terms of improvement of nutritional quality in maize. So with this, I thank one and all for your patient hearing. I shall be thankful if you can have some questions or any queries if you have. I'll be very happy to share with you.
Thank you, sir. After the valuable session by our Dr. Firoz Hussain sir, we are sure that all the participants have benefited a lot. This benefit has encouraged them to know more, and they have posted some important questions on the YouTube and Google Meet. So here we go with the questions, sir. The first question is, sir, does the rise in the level of nutrition increases the probability of pest infection?
And not really, because these nutrition level is increased only in the grains, and there are of course the varieties which we develop. They side by side is also resistant to resistant to diseases. So it's not that we are developing nutritious varieties and it becomes susceptible to pests. We take care of that, but then there is no relationship as far as these traits are concerned, which I have mentioned. That if we increase this such iron zinc, it doesn't even make susceptible to such diseases. So absolutely, it's not a problem.
Okay, sir. Sir, here the second question: what much extra effort need to be amended to meet the level of agricultural development of Israel? It is posted by Samira Siddiqui. Hello, sir.
Yeah, Israel is one of the pioneers of certain areas of agriculture. They are very, very advanced because water is scarce. Water scarcity is a major problem. So they have such a technological advancement because of the fact that their crop varieties are water efficient, and they give water so efficiently that not a single drop is wasted. So Israel has its own advantage, but definitely I'm sure biofortified crop varieties. In fact, I just only made about these. Now there are 71 biofortified varieties across rice, wheat, millets, vegetables, and for horticulture have been developed. Oilseeds, pulses have been developed by ICAR. So, of course, it's any intergovernmental issues. We'll be very happy if such biofortified crops can be grown there in Israel. So the whole country or any human mankind should get benefit out of that.
Okay, so sir, last question. Yesterday, some people posted about the CRISPR. If CRISPR gene get mutated in target patient, can it create a deadly effect?
I can only talk about plants where now CRISPR is a very powerful technology. What we
Yes.
do is, you see, the breeding method takes almost, which I did not mention. In fact, it takes 10 to 12 years to
Oh,
develop a variety. It's a pretty
okay.
long time. And
Oh,
but of course, we have the advantage of molecular breeding, which I didn't touch upon, where we look at the DNA and profile it. So we cut short the cycle of 10 to 12 years to almost 4 to 5 years. Then CRISPR is much shorter cycle. You don't have to cross. You have to only take a variety and mutate that gene by simply removing one or two nucleotides, adding one or two nucleotides. So far, there is no report of any adverse effect as far as plant is concerned. If you mutate the gene through CRISPR, it's a very safe technology as far as maize is concerned, as far as crop plants are concerned. In
okay.
China, I must mention, Chinese breeding program have now adopted this CRISPR technology in a big way. So what they are doing, they are taking their hybrids and they are mutating one or two genes because it's a very targeted. And let me suppose they have a very good hybrid, but it is disease susceptible. So they just mutate one gene using CRISPR, make it resistant. So it's just a 2 to 3 years program. That is what it is.
Okay, sir. Thank you, sir. This question answer session was really enlightening, informative, and interactive. We are grateful to Dr. Firoz Hussain sir, who has given us an informative and insightful talk on nutritional quality enrichment in maize through genetic avenues. And I am quite sure our participants have really benefited a lot. We would like to thank you for taking time out of your busy schedule for this webinar. Once again, thank you, sir.
Thank you. Thank you very much.
Thank you, sir. Now, I hand over the proceedings to Dr. Nikhat Naqvi, Associate Professor and Head of Botany Department, SFS College. Over to you, Madam.
Thank you, Dr. Susan John. Good morning, everyone. This is Dr. Nikhat Naqvi, Associate Professor and Head of the Botany Department, St. Francis de Sales College, Nagpur, welcoming you to the second session of today's national webinar on recent developments in genetics and their impact on religion and society. For this session, we have renowned academician and researcher par excellence as our guest speaker. It's my pleasure to introduce our esteemed guest speaker, Dr. Tri Srinivasu sir, Professor of Botany and retired Head of Postgraduate Teaching Department of Botany, RTM Nagpur University, Nagpur. He has vast teaching experience. His area of research include mutation breeding of oil crops, digital biodiversity, electronic herbarium, and digital database preparation of wild and medicinal plants, molecular taxonomy and phylogeny, tissue culture, plant identification on molecular basis, and water pollution. He has completed 5 research projects. During his tenure, he has been instrumental in carrying out FAST program by DST and UGC SAP. He has conducted training programs in very significant research fields of e-herbarium and digital database preparation, DNA sequencing in plants. His DNA sequence submission to GeneBank includes Phyllanthus and Hygrophila species with accession number. 8 students have been awarded PhD under his able guidance. He has published and edited many books on flora and biodiversity. He has been co-author and editor of Nagpur Raj Bhavan Plant Biodiversity by RTM Nagpur University. He is life member of various prestigious academic societies like Indian Science Congress Association, Indian Botanical Society, All India Angiosperm Society. He has organized number of workshops and national conferences, refresher course in life sciences. He has published 63 papers in various national and international journals of repute. He has chaired number of technical sessions in various national and international conferences workshops, and seminars. He has also presented 82 papers in different conferences. During his illustrious tenure, he has shouldered many academic and administrative responsibilities. He has been member of RRC in Botany, Chairman Board of Studies, Botany, RTM Nagpur University, member of Board of Studies in Molecular Biology and Genetic Engineering, member Academic Council, RTM Nagpur University, Nagpur. I now invite our guest speaker of today's session, Dr. Srinivasu sir, for his talk on a very important and relevant topic which has affected life of each and every individual since almost a year. And he will be deliberating on SARS-CoV-2 and its impact on religion. Gentle reminder to all the participants to post their questions in the chat box along with the names and affiliation. Dr. Srinivasu sir, I request you to enlighten our audience with your presentation.
Thank you very much, madam, for inviting me for the 2 days webinar on national development in genetics and its impact on the religion and the society. And I thank Susan madam also for specially talking to me and inviting me and to participate in this particular 2 days national webinar. Thank you very much, madam. Thank you. Coming to the topic. I have chosen this particular COVID virus today because this is the current problem we are facing and we are solving this particular problem today. And to some extent we have successful, and that's why to enlighten all the audience of this particular 2 days webinar, I have chosen this SARS-CoV virus 2 and its impact on religion and society. What is COVID-19 virus? The scientists named this as a SARS, that is called severe acute respiratory syndrome virus, and it is associated with what we call coronavirus, that is called SARS-CoV, and is called as a SARS-CoV-2, and that causes what we call COVID-19 infection, and it was first identified in Wuhan, China. Now there are several species or strains of this coronavirus that can infect human beings, and 4 of them are very common and they cause what we call common cold. And there are 3 other coronaviruses which cause a very severe infections, that is called SARS, Middle East Respiratory Syndrome, COVID-19. And now the entire globe is suffering with what called COVID-19. Now the World Health Organization, WHO, declared it a pandemic as it spread to nearly every country in the world. So how it is going to spread this particular virus? The virus are airborne and spread through the close human contact and in the droplets from coughing and sneezing. It also transmits through hugging and kissing, sharing utensils for eating and drinking, and speaking to others within a distance of 3 feet, and touching someone directly also. This way it is spreading. And what are the symptoms? Whether the particular patient is infected with this coronavirus, these are the what you call the symptoms. Mostly very common symptoms like fever, chills, cough, shortness of breath, difficulty in breathing, sore throat, congestion or running nose, fatigue, headache, body ache, or sometimes a loss of taste or smell, nausea, vomiting, or both, and diarrhea. Now there are some jokes based upon this particular loss of taste and smell. So husbands claim that wife food is not a tasteless, is a tasteless. So she says, if you say is the tasteless are not good, I will complain you are infected with corona. So it is a simple what you call joke, which I have seen in some newspapers also. Most people develop pneumonia, but while some other, mostly the older people above 60 years age, had a what you call a long-term damage to their liver, kidney, and lungs. Higher risk of developing serious illness in older adults and people of any age with underlying health conditions, particularly if they're what you call facing some problems. like heart disease, stroke, diabetes, cancer, high blood pressure, and obesity, and particularly the pregnant women. So how to prevent this particular one? Now governments, several governments in the world, they are what you call facing several problems and giving a lot of precautions to the public how to prevent them. So one of the important thing is washing hands frequently or cleaning with an alcohol-based detergent. Nowadays, there is a lot of sales on the what you call the hand sanitizers and alcohol-based detergents. So they are available in the market very easily. So by using them, we can prevent particularly the COVID virus. And avoiding touching the eyes, mouth, and nose, these are the places where this COVID virus can enter. So we should avoid these places with unclean hands. Covering the mouth and nose with a tissue when coughing and sneezing. So to avoid this particular one, the COVID virus, nowadays people are recommending using the mask. And the another point is that avoiding the sharing of food, drinks, and utensils, particularly the home quarantine. Here is important because one can avoid the food or drinks or utensils of the infected people in a particular house. Staying at least 3 feet away from other people, that is very important. So if you are talking to somebody else and maintaining distance of 3 feet, and you can avoid this particular one. Otherwise, what happens? Whatever the droplets which are coming to the mouth or whatever the nose, or while sneezing or coughing, so they will what you call infect the other person opposite to that person. So regularly cleaning the surface with disinfectants, we can avoid this COVID virus. Though thousands of people have infected, but still millions of people are safe because they are following all these prevention activities. Now, how many types of coronavirus we know today? So, coronavirus belongs to what we call subfamily Coronavirinae or family Coronaviridae, and this is what we call classification. And there are seven types of viruses which infect the human. Of course, in the animals there are some other viruses strains are available, but in case of the human, types of strains which infects the human being, and 4 are very common, particularly 229E. That is the alpha coronavirus, which causes a cold. NL63, it is also called as a alpha coronavirus, which also causes a common cold. OC43, HKU1, these are the 2 beta coronaviruses, and these 2 also cause what we call common cold disease in human beings. But the rarer strains are available, and these rare strains that cause what called more severe illness includes MERS-CoV, which causes the disease called MERS, and SARS-CoV. The virus is responsible for what we call SARS disease. And the third one, the most important one, which is a pandemic today, is a new strain of virus that is called SARS-CoV-2 that started circulating and causing the disease today is called COVID-19. Now coming to the, before going to the other details, we are going to study this particular here, the structure of the coronavirus. As I am giving very simple structure and details of this particular structure, a common man, a layman can also understand this particular structure, how it looks. So I have prepared some figures here, and this is what we call on the left side, how this particular coronavirus looks under the electron microscope. So you can see here, these orange color or red color, you can say these are the spike proteins, and they are present on the membrane. Now you can see here, this is the membrane. On this membrane, you can see these are the spike ones. These are the spike proteins which infects or causes what we call symptoms. And here, inside this particular membrane, inside this membrane, you can see this is what you call it genetic material RNA. So this is a helical structure you can see. Now the same thing you can observe here. This is what we call a membrane, which is made up of glycoproteins, and these are the spike proteins, and inside this is what we call RNA, the genetic material. Now this particular virus will come into contact the membrane of the humans. This is what we call human membrane, having some receptors. These are called ACE2 receptors. Through this particular ACE2 receptor, this particular virus come in contact and enters inside the what we call human cell. There are certain enzymes which are required for synthesis of the receptors, etc. So this angiotensin, a converting enzyme that is called ACE2 receptor, through this particular one, it enters, and there is what you call a transmembrane protease serine 2 is an enzyme that in humans is encoded by what you call TMPRSS2 gene. And now we will come to details of what is the role of each receptor and what you call other enzymes which are involved in entering into the human cells. So this is the structure of the coronavirus. Basically, I'm giving, and now you see this genome of this particular one, and they have been sequenced within a 20 days or 10-15 days time. The RNA genome was sequenced, and the total length of this particular genome is a 29,881 base pairs in length. Now, if you see the gene bank, the accession number is given. There you can see the details of this particular RNA DNA sequences. And this particular small piece of RNA, which is encoding nearly 9,860 amino acids, that is a proteins. The spike proteins are also produced by this particular virus having this particular length. Now, here, how we come in contact with this coronavirus? That is what we call binding of spike proteins to the receptor human cells. As I have shown you earlier, these are the spike proteins. How these particular spike proteins are. You can see here in this particular structure, this is a spike protein which is present on the membrane of the coronavirus here. And these spike proteins come in contact with the receptors of the human cells. As I have shown you, these are the receptors, ACE receptor, etc. You can see here. So these receptors are here, and this particular spike protein will come in contact with these human cells through the receptors. Once they come in contact, now you see this spike protein structure also very clearly. This spike protein is made up of two subunits, S1 and S2 subunits, and there is what called RBDO, right? Through this particular one, it will bind to the receptor human cells, and this viral spike may try to enter into this human cells through this receptor. This is the structure of this particular one. We will see what is the spike protein. The spike protein of SARS-CoV-2, which plays a key role in the receptor recognition and cell membrane fusion process. Now you can put in a question, why this coronavirus is not infecting the other animals or any organisms, only human beings? Because this is a what you call the spike protein, which plays an important role in recognition of the human cells through this receptor. And after recognizing this receptor, and this what we call cell membrane fusion process takes place. So this spike protein was sequenced, and it is composed of two subunits, S1 and S2. I have shown you. Here you can say S1 and S2 proteins are there. And the S1 subunit contains a receptor binding domain that is called RBD. This is the RBD, receptor binding domain. And this receptor binding domain will what you call bind to this particular receptor that is called ACE2, receptor angiotensin converting enzyme that is called ACE2, while S2 subunit mediates the viral cell membrane fusion for forming a 6 helical bundle via 2 heptad repeat domain. So now this particular one is binding to this particular human cells and making a fusion. So these are some of the what you call bioinformatics figures I have shown you. And here this is the host cell, and this is the SARS-CoV-2 virus having these spike proteins. As you have seen, this is a S1 and S2 subunits and RBD, receptor binding domain. And now the host cell, human cells, having the ACE2 receptor, and to this particular receptor through this RBD domain, it is binding to this particular one. And you can estimate the patient is infected with the virus, coronavirus, how much that you can estimate by small test that's called antibody, and that reaction So it is showing the illustration of the human antibody latching into the receptor binding domain RBD of the spike protein of SARS-CoV-2. And this is what you call, they have shown you the UNSC School of Medicines. Now coming to the how this particular virus multiply in human cells. So in simple figures I have shown you. This is what we call the coronavirus with the RNA inside the genetic material and the protein code with the all spike proteins. And these are some of the receptors present on the human cells. And this will come in contact the receptors and they make an entry inside the cell of the human cell. Now inside the host cell, this what we call a virus has made an entry. And after that, the protein code is dissolved and this RNA will multiply. That is what we call replication. You call, of course, a lot of details are available, but I'm not going to discuss here the replication. And the copy of this particular RNA which enters into the host cell will multiply and make several copies of this particular one. At the same time, the RNA having some portion of the genome which is meant for production of the what you call spike proteins, etc. And that portion of the RNA with the help of the ribosomes and the enzymes present in the human cell, that the host machinery is used for synthesis of the spike proteins and the protein code, etc. Once this protein code with the spike proteins are prepared, the duplicated or replicated copies of RNA enters into this particular one, and the fresh virus are synthesized, and they will leave the human being and try to contact another human being through the transmission. Now you can see here, this is the particular virus. These are the antibodies reacting to this particular one. This is the word simply antibody neutralizing the virus. If anything enter in our body, any foreign body enter in our body, our antibodies try to bind and neutralize those effects. But if the what called the virus population is more, yes, automatically if this percentage is more, and the person will show the symptoms of the virus. Okay, coming to the spike proteins, as I told you, this is the RNA genetic material of the virus, and here this is the portion, a dark blue color one is the what you call a region which is capable of producing the spike proteins, right? This particular what you call cut off, and using some enzymes we can cut off and produce what you call a small piece of RNA, and this small piece of RNA which is capable of producing the spike proteins. This is what you call RNA coding the spike proteins, and the sequence I will show you in the next slide. And these what you call the spike proteins producing RNA region are incorporated into the lipid nanoparticles. Once, of course, now people are preparing the mRNA vaccines. How they are prepared? Now this portion of the RNA which is amplified and inserted into the lipid sac-like structure that is called lipid nanoparticles, and the RNA which is inserted in the nanoparticles can be used for the vaccination program. How you can see, this is the roadmap of mRNA vaccine development. Now the first report of respiratory virus outbreak in Wuhan, China in December 31st, 2009. Of course, I am highlighting some points, important points. And now within 10 days, the 2020 January, the sequence, the what you call a coronavirus sequence, the RNA sequence was published. So the RNA is isolated and sequenced, and that is established. There are several variations are there. Several virus strains are available. Whatever the strains available, they have been sequenced here in this case. And within 3 days, of course, people have decided how the spike proteins are produced, which portion of the RNA are responsible. And these are the particularly VRC and Moderna, the companies, they have decided mRNA 1273 sequence, and that is responsible for the product symptoms and everything. Then immediately, the next day onwards, they have started producing the what you call initiated the production of cGAMP production, and the in vitro expression of the mRNA, which is isolated for the sequence, everything has been done very fast. Hardly few weeks time, they have done all these things. They have started vaccinating the mice to find out the particularly the effect of vaccines on the mice. in February 4th, 2020. Now they have what you call synthesized this particular drug and they want to test it. So they have confirmed very short period of time. You can say today now in March 16, 2020, the phase 1 clinical trial started, and today now you see the clinical what you call trials second phase is also completed, and the what you call the vaccines are available for vaccination mass vaccination program in the world, particularly in North. Now, this is what you call a roadmap of the mRNA vaccine development, and this is the spike protein structure which was deciphered later on. Now, these are the two important companies in America and USA, UK, the Moderna and Pfizer BioNTech. These are the two different companies. They have developed vaccines based upon the mRNA, particularly which codes for the spike protein. I have already shown you in the earlier structure the spike protein, right? And this is the region. So with the help of this particular one, they have started synthesizing vaccine and undertook some trials, phase 1, phase 2, and confirmed. And the permission has been given to mass production of this particular mRNA vaccine for the first time in the world. Earlier, nobody has given permission for the mRNA vaccines. And during this pandemic period, as an emergency, the FDA, the Food and Drug Authorities, they have given the permission to test clinical trials, and they have given permission for the mass vaccination of this particular one. Now, the only difference between the two vaccines are here the mRNA they have used, but here there is a slight difference in the lipid nanoparticles. Some people have used the small nanoparticles and some people have used the bigger one. Because of this variation, the storage, the temperature for storage of this mRNA virus requires is slightly different, because this particular mRNA is very fragile and it cannot be stored in a normal room temperature. So we need a very minus 35 to minus 70 degrees temperature to store this particular vaccines. So the slight different in a lipid nanoparticles means slight different in storage temperatures. Now let us see here what are the different ingredients which are used by this Moderna and the Pfizer BioNTech. These are, of course, mRNA, which is common in both the vaccines, and the lipids, different categories of lipids combinations, you can say, they have used. And all these are the ingredients which are used in the Moderna vaccines. In the Pfizer BioNTech, they also prepared based upon the mRNA, right? But the ingredients are slightly different. But because of this particular one, the storage facility varies. That is very important, because these facilities are not available much in India. So that's why we have gone to a different categories of vaccines. Now, this is what we call, I said, the mRNA sequence, and 1273, the base pairs of which is responsible for the spike proteins, is a part of this particular one. Here you can see this is the sequence of the mRNA, the coronavirus. These are all. Sometimes you can see here the guanine, adenine, all these things are there. You can say pseudo nucleotides are available. It means that in this place, anything can come out of the 4 nucleotides present. Any one can be inserted, and even that will fulfill. So this is the what we call sequence of mRNA. And of course, those who biology students can understand, this is the protein coding region on the both the sides of 5' and 3' UTR, and there is a cap mRNA which is released into the plasm, cannot be stable, and it will be degraded with the enzymes. To protect this particular mRNA from all these enzymes, we have a cap on one side that's called 5' and what we call the poly A tail. A tail is there on the 3' end. So this is what we call the structure of mRNA. Now coming to the mRNA vaccine platform. This is the idea. This is the spike protein, what we call a therapeutic protein. And generally, this protein, which is synthesized in the what we call cell, and what are the different amino acids present in the spike protein? Here, this is the what we call structure of this particular spike protein is sequence, and these are all different amino acids present. So this is amino acid sequence, And if you know the sequence of amino acids, you can deduce particularly the mRNA sequence very well. If you know the mRNA sequence, you can study what are the amino acids which are going to be coded. So decoding this particular amino acids by using the computer, that's called computation in the bioinformatic practical. So we conduct this particular thing, and students can understand very clearly the what you call mRNA sequence of this particular amino acids. And now this you put it in a what you call plasmid virus, a circular plasmid DNA virus, and this is what you call the DNA sequence. And after adding this particular one, you can break down at this point, and you can see it will be a straight linear molecule. And by using the what you call enzymes which are required for this particular one, nucleotides, you can sequence this particular one, and you can purify this mRNA, and this purified this portion of the what you call RNA, which is going to what you call code for spike proteins. So they are inserted into the lipid sac molecules. So this is the mRNA, and this is the left what you call lipid sacs and nanoparticles, and this is a which is inserted inside, can be used for preparation of the what you call vaccines. A formulation, which is of course I have given already the ingredients of the lipid molecule mRNA, which is common in both these cases, but the lipid sac structure varies, and this is because of different ingredients they have used by the two different manufacturer, Moderna and Pfizer, etc. And a mass production and vaccination program, you can do this particular one here. Now, when the vaccines are injected in our body, automatically against these vaccines which are having the mRNA and this lipid sac, the what you call antibodies are produced, and they try to attack and produce a large quantity, and they try to be ready. If the really coronavirus infects your body, it will try to attack. That is the reason. So what you call here, this is the human cell, and these are the what you call coronavirus. If they try to enter inside this particular one, these what you call antibodies which are trying to. So if this particular virus which entered in our body and try to multiply and produce several number of what you call viruses, and this is the receptor, you can say, and automatically this particular spike protein, this is spike protein, if they are available, immediately the antibody will come and attack this particular one. This is a host cell, that is human cell, and nucleus and cytoplasm containing ribosomes and mRNA, right? This particular vaccine, which is neutralizing this particular what you call virus. Now, the first time I told you the RNA vaccines are synthesized and the permission is given to them first time in the world. And what are the advantages of this RNA vaccines? So it is very easy to manufacture because you have isolated mRNA, a small piece of RNA, and you can multiply and you can produce a large quantity, billion doses of these vaccines you can synthesize very easily in the laboratory. And it's very inexpensive because multiplying a particular copy of the mRNA, hardly it is of 1273 base pairs length, and is very inexpensive, and we can produce a large quantity. That is another advantage of this RNA vaccines. And this process does not require what you call a pathogenic infection virus. We don't need any virus. In the other cases, the vaccine generally are prepared using the same pathogen, right? The pathogenic virus, and we prepare the vaccines. But here we are not using the what you call pathogenic virus. We are taking a part of the RNA, which is what you call causing the symptoms, and that process involves only this mRNA small piece or segment and not the entire infectious virus for the production of the vaccines. Now, as I told you, RNA is a very delicate thing. You cannot store this particularly under normal room temperature. So this is the RNA is not a what you call a super temperature stable. So we need a lipid sacs to carry this particular mRNA molecule into the cells, and we are putting this mRNA in a capsule-like structures that is called lipid nanoparticles, and we store them. Currently, there are 2 RNA vaccines are available, as shown earlier, and out of this particular one, they have tested the effectiveness of this particular RNA vaccines. 95% effectiveness in preventing COVID-19, and they are approved, accepted, and permitted to production in a mass scale in USA. And now in USA, they are using these mRNA vaccines. But in case of India, we are using the two different Covaxin and Covishield vaccines. So India has approved two vaccines that is called Covishield and Covaxin. Covishield has developed by the Oxford University scientists with AstraZeneca, and particularly using the Indian variant that is called a virus, a strain is called AZD12221, and this is mass scale production by the Serum Institute of India, Pune, and that is manufacturing and supplying for mass vaccination program. And the Covaxin is another one, which is also indigenously developed by Bharat Biotech in Hyderabad in collaboration with the National Institute of Virology and ICMR, Indian Council of Medical Research. And now these two vaccines are used for a vaccination program in India. So the Covishield vaccine has been developed by using the virus adenovirus. What is the difference between the Covaxin and Covishield? The Covishield is used by using the adenoviruses. Adenoviruses is common, which produces a cold and cough and other problems in particularly the human beings. That's called adenoid related virus. They are called adenoviruses, and they cause only cold, something like that, but they are very safe. The Covaxin, which is a virus, which is a vaccine, is produced by using the what you call the inactivated, what you call coronavirus. So the dead or inactivated coronavirus is used for synthesizing Covaxin virus. Okay, but then you may have certain doubts about the vaccine. Sir, you are using the mRNA, whether we may get this particular mRNA incorporated in our DNA. Certain doubts are there. I'm clearing those doubts also. Do you have a doubt that RNA vaccines change our DNA? No. The RNA in the vaccine does not enter into the nucleus, interact with our own DNA in any way, and it is quickly degraded. So it will not go inside the nucleus of the human cells, and it will be degraded in the cytoplasm itself. Will they be effective against the new viral variants? How easy will they be to change and update? Most likely, whatever the variants, mutation states are coming up, they are also nullified and ineffective. Particularly, if you find any mutation, and they are also what you call not going to affect. Particularly, they have seen lab results have shown us the Pfizer and BioNTech vaccines that is neutralizing what you call N501Y mutation. So this is the one mutation a variant has developed, but that is also suppressed with the same vaccine. Now, can you get a COVID from these vaccines? We are not going to affected with the COVID disease with the introduction of the mRNA vaccines. Why do these RNA vaccines require two shots? Because the one shot is to what you call immunization, and second shot is called a booster for improving the immune system in our body. Then second, you can expect why do these vaccines need to be kept in a so cold? So in the newspaper, you must have read it, particularly the storage problem in India. And that's why we have gone for Covaxin, Covishield instead of mRNA vaccine. Because the RNA is so fragile, it can be easily break down at a warmer temperature, room temperature. Even water can break down this particular RNA background if it is not stored in minus 35 to 70 degrees centigrade. That is a very difficult in India because India is a warmer country, and we storing this particular one, we require minus 80 degrees refrigerators, and it is not available everywhere. That's why we have used the Covaxin and Covishield instead of mRNA vaccines. Now, what is the impact of this particular COVID virus on the worship places and worship styles? Because of this particular COVID virus, almost all worship centers are closed. Churches, temples, mosques, gurudwaras, everything, entire globe are closed down because of the COVID situation. And several festivals with enthusiasm, they are also stopped. Cancellation of events during the festivity, cancellation of processions, congregations, prayers, suspension of the all divine services, events, rights, restricted permission to attend even marriages, rights, etc. worldwide, even today also. There is some restrictions. So they increased vigilance regarding the cleaning worship areas. Of course, as because this COVID virus has become a pandemic, so the worship places are what you call cleaned daily with a lot of care, and particularly providing the what you call hand washing stations. Even in the colleges, any worship places, any public places, the hand wash stations are at to clean, wash your hands before and after acting. So and not only this particular one, even the worship places, so members are using to what you call maintaining the social distancing and preventing measures. A lot of propaganda has been going on how to prevent or avoid this particular COVID virus. Because of this particular one, so avoiding the gathering as a communal iftar and suhar meals and reported change in a what you call worship styles, and people are going for religious what you call it, online what you call, I think it has gone to next page. So people have changed their worship styles and religious habit due to the pandemic, watch the religious services online or through TV instead of in persons, and started digital online services, live streams with the charts, chanting songs, bhajans, etc. Some priests began offering drive-through confessions, day of prayer and fasting, etc. So these are some of the changes the COVID virus has brought changes in our worshipping styles. So with this, because the time is almost over, the allotted time is almost over, and we can discuss, interact with some of the doubts if you have. Thank you very much for giving me an opportunity to discuss with this particular the structure of the coronavirus, the preventive methods, the vaccinations, particularly the mRNA vaccines, which are newly introduced first time in the world, and the normal vaccines, and how this particular virus, a very tiny particle, has in fact affected particularly the worship places. Thank you very much for giving me an opportunity to explain this particular. Thank you.
Thank you, sir, for your insightful presentation and informative talk. It is indeed an honor to have you with us as guest speaker. Your deliberations are so relevant in this pandemic times. You have explained in detail different aspects of SARS-CoV-2 in a very lucid manner, including its structure, spread, and also precautions to be taken. You have also explained very nicely the roadmap of mRNA vaccine development. And due to this virus, we all have gradually adapted ourselves to the new normal. And vaccines, they offer ray of hope, but there are a lot of doubts in the mind of the people. So you have convincingly cleared a lot of doubts about the vaccine being administered right now. Sir, your talk has generated a lot of curiosity and excitement among the participants, which can be gauged from the number of questions and also wonderful comments coming in the chat box. And with your kind permission, may I take a few questions via participants' questions?
Thank you, ma'am. You can ask me.
Okay, sir. So first question I would like to take up is there any alternative transmission route for COVID-19 and SARS-CoV-2?
No, madam, only this way. The receptors binding proteins, they will bind the receptors on the human cell, and through that only they have to enter. Even though this is a very tiny particle, the entry inside the membrane is not possible by another way.
Okay, thank you so much, sir. Another question that I would like to take up. Will the virus virulence change during the pandemic, and what fate awaits SARS-CoV-2?
That I explained you in the replication cycle, how it is multiplied in our body cells and they spread. That is, I have explained you, the virulence stage only, this will happen. Because the mRNA which is released and multiplied and enter, what we call is synthesize, using our own machinery, the human machinery, and they multiply and they spread. So the vaccines will, what we call, make ineffective by that way. The viral
Yes.
stage, that is the one stage.
Yes, sir. Sir, there is a very interesting question, and I think for this we will require another session. The participant, Dr. Samira Siddiqui, is asking, with your knowledge and experience in medicinal plants and the role of medicinal plants in traditional information about medicinal plants, what can be the scope of medicinal plant research in prevention and controlling of this virus? I think this will require another one session.
Work is going on, madam, and people
Yes,
have
sir.
suggested what you call the immunity improvement. There are certain things, ginger, all these things people have suggested, but the
Yes.
clinical trials have not been conducted. It requires some more time to carry out and finalizing this particular one. So whatever the suggestion, generally for common immunity, what are the ingredients we use? Those ingredients you can develop a resistance. That's all. And work is going on.
Yes, sir. Thank you so much, sir. On behalf of the management and principal, I am immensely grateful to our guest speaker, Dr. Srinivasu sir, for accepting our invitation and delivering a very informative talk on the current pandemic, which everyone can relate to. Thank you so much, sir.
Thank you, madam. Thank you.
Okay. I now hand over the proceedings to Dr. Susan John. Over to you, Dr. Susan John.
Thank you, madam. I am delighted. Now it's time for the third session. I am delighted to introduce Dr. Victor Ferro to everyone. Father Victor Ferro holds a doctorate in philosophy of science with specialization in science-religion dialogue from Philosophy Department of Jnanadeep Vidyapeeth, Pune. He is the Dean of the Department of Philosophy and co-ordinary of the Special Study Center of IGNU at Rachel Seminary. He has authored 5 books and has edited a book of proceedings of Annual Conference of Association of Christian Philosophers of India. He has several research papers published in peer-reviewed journals as well as edited books. He is the founder director of Science Religion Sangam Goa and is actively involved in science and religion dialogue. He has also organized national and international conferences and presented research papers in several national and international conferences. I accord a hearty welcome to you, Father, to this session. We are indeed glad to have you with us today. I kindly request you, Father, to take over. Over to you, Father.
Yeah, thank you, Dr. Susan. I want to share my slides and begin. Can you hear me?
Yes, sir. Yes, Father. We can hear you. Yes, Father.
I'm going to begin now. Can you see the screen that I have shared?
Yes, sir. Yes, Father. We can see your screen. Yes.
Okay, this is the title of my talk now, Ethics of Gene Editing, which is a very important development that has happened today in life sciences, and there are a lot of promises in this field. Medically, also enhancement for human beings. Earlier we used to heal sicknesses. Today we may want to add new features, characteristics, and enhance human life and many other things. It has given also power over evolution. Human beings can now control evolution, the destiny of animals, plants, and therefore it becomes a very, very important. area to discuss ethics as well as the consequences of this science. So my presentation has three parts. I'm taking one important technology of gene editing, understanding what we call CRISPR-Cas9, understanding human flourish. With the second part, where I like to understand will this technology bring about human flourishing, and towards the end, we will take up ethics of care. So ethics of care is about inclusive, not only human being, but will include all forms of life and our planet Earth. And therefore, we cannot simply be having an ethics which is anthropocentric, only thinking about human good, but the good of every life form on Earth is required to be put into the ethical calculus. And that's how we will try to understand the new ethics that is required in order to face the new technological developments in life sciences. So let me begin by a short survey because I have less time and I can't get into this. Perhaps others might have done technicalities of understanding DNA, understanding the bases of DNA, the four bases, and the structure of DNA, RNA, and things of this nature from molecular biology. Central dogma of molecular biology is from DNA to RNA to proteins. This is how we can do genetic engineering. RNA is a messenger, a guide, and it will help us to produce proteins. This is what is happening in the cell, and therefore, if we can manage this process, we will be able to take control over the genetic life of a cell, and we can modify it, we can add features to it, we can silence certain genes, we can also open genes which are silenced. And so for benefit of humanity as well as for animals and plants and microbes, perhaps this control over the DNA is very, very important and very good development. Now, in this context, CRISPR-Cas9 is a very interesting tool of gene editing. It makes the immune response of a bacteria to a viral attack. So, bacteria that which is attacked by a virus has a way of tackling it. And now, once we understand it, we take control over this technology, and we can use it for gene editing across animals, plants, and also human beings, not just on a dish or a test tube where we do the gene editing, but at the same time we can do it in a living cell. So that's where the developments are quite important, and also ethically we need to respond and find ways of addressing these new developments. And so to begin, CRISPR-Cas9 stands for clustered regularly interspaced short palindromic repeats. So that is, there are repeats. Their palindromic means, like one example of a palindromic repeat could be madam. Madam is an English word, but you can also read it from reverse. It repeats MADAM. If you go back, it will be same from the other side. So therefore, there are this type of repeats, and these are interspaced. And what is there in the interspaced neighborhood of the repeats? In the interspaced neighborhood of the repeats, we have the memory of the virus, the DNA of the virus that had earlier attacked the bacteria. And now in this way, this retention of the memory and the genetic memory of the virus, the CRISPR molecular structure becomes a vaccine card, because for a future attack of the virus, it will identify because it will create an RNA where the genetic structure of the virus is there, and this RNA will go and identify, recognize the DNA of the virus. Once identifies, Cas9 is a protein that is produced, it will go and cut the virus. So once it is like a genetic scissor, Cas9, it cuts the virus at certain points in the structure of DNA already identified by RNA, and therefore the virus is disabled, and this is how the bacteria dealt with the virus. It has evolved through its process of responding to viral attacks. So CRISPR-Cas9 therefore becomes a very powerful editing tool. We are only mimicking the bacteria in its editing. In other words, in cutting the DNA. So it's like we can identify a particular strand of DNA. If we want, we can cut that using CRISPR, cut that with Cas9, and we can also put a desired DNA in its place. And so in many ways, there are certain sicknesses which are genetically induced, could be addressed, and that's why we can actually repair DNA which is already sick, or we can target even single alphabet of a DNA, and this is how this becomes much easier than before, because earlier it was more difficult. We had to use viruses to be vectors to carry our RNA messages and things like that, to edit genes or DNA. So RNA is like a GPS, which is going to target DNA strand that we want to sort of address, cut, and it is like cut and paste that we do on the computer, cut and then paste the desired DNA that we want. Or we can also wait, because there is the repair mechanism of the DNA, and it can also by itself produce another DNA strand. But that is a bit dangerous, because it can lead to genetic changes which are undesirable and we don't know and we may not be able to control. So therefore the best would be to paste the desired DNA, the good DNA, so that the sick person his sickness is addressed. Now in this case, I was already talking about therapy, that is about sick people getting therapy, but that is also another thing, like we can also enhance not just therapy, but we may try to engineer certain genes and introduce them of course in the very first stage, in vitro. Perhaps we can design babies, we can design humans the way we like. So there is gen-rich generation. So genes will be enriched in them, and they can become post-human, transhuman. We don't know exactly what it will take and where it will reach. So these are things that are happening about human beings for enhancement. We can also do the same with animals, plants, microbes, and therefore for the first time we are having power over evolution. Nature had power over evolution down the ages. Right now it is humans, science and technology is taking power, over evolution and may lead it and guide it to different to make a difference to this planet Earth. We may even help those extinct animals to come back and things like that. But at the same time, there are good sides as well as bad sides, as we know of any technology. It can be abused. It can be things can be very harmful to humans, and at the same time, we can also harm other lives on this planet Earth. That is the reason we need what we call human flourishing and ethics to look at this. Now, I like to go to human flourishing of Aristotle because it allows me, not just to react to this science as if we are playing God or, in a very negative way. It is very dangerous, so we will not do it. So those type of reactive responses, which are taken sometimes given ethical color, are not actually good in this context. We cannot be so afraid of technology. We cannot show technophobia, but we need to be courageous. We need to understand the good is yet to come, and we need to understand how it will unfold and be in control of the use of this technology. Therefore, Aristotle's Ethics, his book Nicomachean Ethics, where he wrote this ethics to his son, where he talks about eudaimonia. Eudaimonia is translated many ways. Some people translate it as happiness, but in many cases it has been translated as human flourishing or excellence. It is fulfillment of one's nature, fullest actualization of one's potentials. Therefore, fullest actualization, what we can do. As a scientist also, we can actually fullest actualization of even this technology results in kind of excellence. And so we become, if we talk from religion, we'll become co-creators with God. God, who is the creator, we participate in the creative work of God, and new genesis and a new world could come through this technology. So therefore, it has a teleological character. It has something to tell us where we are heading, and it can give us full realization of potentials which are there in animals, in human beings, and in everyone. But with this knowledge, this is not enough to let everything become reach its fullness. That includes humans. Of course, Aristotle is talking only about humans. Therefore, he is anthropocentric. He is not concerned about plants, animals, anyone to sort of actualize their potentials. When he talks about this ethics, human responsibility is to become more and more fully human. And so it involves pursuit of virtuous life. We cannot actualize the potentials without virtues. So living virtuous life through practical wisdom, what he calls phronesis. Theoretical wisdom, theoria, is Sophia, and that is basically philosophy, knowledge based on reason, reflection. Here, it is more reflexive wisdom which is reflexive in action, in praxis. While we work with hands, or while we work, we are able to, live virtuous life and bring excellence. So we need a habitual disposition, in Greek called haxis, to act according to practical wisdom, which is not that it is not something like, you reflect and act, but in the very act, there is this wisdom embodied inside us, and we are able to become living virtue in our dealing and working every day. So eudaimonia therefore involves arete in Greek, virtues, and phronesis. These are the two things. But the problem here with Aristotle is, this is a very beautiful way of being human, but at the same time, it is very subjective. Each person has to live by himself the virtue, set of virtues, and which is also anthropocentric, only human centric. It doesn't address others. How should I be virtuous in relation to plants, animals? For us, how can I be virtuous as researcher in gene editing, as well as one who is in ethical commissions to regulate such scientific technology? So therefore, it is not intersubjective, but simply subjective. That's one problem that we need to overcome. It is deliberative, not intuitive. In a sense, though I have said it comes embodied, but at the same time, it doesn't come without, cultivating, without simply thinking. So there is need of this side, as well as deductive. Deductive would mean we predetermine what is good. Now, this problem that we are facing with gene editing is a developing science, technology, and its benefits, and good is at showing itself case by case, and that is our inductive process where science begins studying case by case and then comes to the final conclusion. So in some way, I thought our ethical response should be one of inductive process, one that will accompany, one that will understand the journey of this science, its impacts on humans, environment, animals, and our planet Earth, and plants, everything. So what could be kind of an inductive ethics? Inductive ethics is there. Most of the ethics is deductive. You think that, okay, this is good, therefore you apply it from a predetermined idea of good. But there are also people like Eugen Habermas, whose discourse ethics through communicative ethics or communicative action can be a good candidate to think. But I have also a problem with his understanding of communicative ethics or communicative action, what he calls. That means we sit together, we decide, deliberate in a free manner, we debate, we contest, and then arrive at what is good. So therefore, it is arrived in a consensual manner. A consensus is arrived, and all of us using reason, we come there. So it is based on the force of reason that let us just imagine that committees or commissions or ethical commissions which are sitting to understand this new science and are trying to reason and find what are the best practices and what can be avoided and what has to be fostered and regulated in this way. So therefore, coming together using reason, coming towards a common consensus, this is what the good that is arriving, to arrive. And therefore, we embrace this method, and by which ethical commissions control or regulate these technologies. So this is one ethical approach. But I would like to, again, expand this ethical approach, because ethical approach to bring care into it, because care, which I started with, virtue. So that type of deliberation, we sit together, we discuss, we debate, but all animated by care and virtue, and to make this communicative action an action of care, so that all of us care not only for human good, but good of all life forms, as well as our planet Earth. And therefore, reason alone is not enough. Habermas is reason alone approach. That reason alone approach can be instrumentalized to suit vested interest as well as lead to what Max Weber calls iron cage of rationalization of society or bureaucratization of society. Because when society is rationalized, like we keep time of flights, we keep time to railways, we keep time to talk, the whole society is rationalized, bureaucratized, and we miss something important in an iron cage of bureaucratization. That's what this man, Max Weber, sociologist, says. But more that is instrumental reason. Instrumental reason is one that reason is instrumentalized and think about only means. We think and we forget whether our goals are right. We don't want to talk about goals. We only want to say, let us say, well, I important. How becomes very important. How do you accumulate wealth and how do you do this? And so, this instrumentalizing reason for means alone, for ends. Means do not justify our end. That's been said quite often. Okay, bad or good means goals have to be always good. So in some way, instrumentalization of reason has to be avoided. Therefore, reason alone is not enough. That's why I thought care, where Heidegger talks about Sorge, the very being of a human person is care. And if we are care, if we are able to care for each other, care for the earth, care for time for science to arrive. So that assists us to come to the application. This is what I would say. Inductive ethics will help us to come to the application of this complex technology, which is going to change us, our lives, our food, our health, our environment, so many things around us. So we need a new ethics, and I'm trying to propose a combination of subjective Aristotelian eudaimonia, but make it intersubjective using Habermas. But at the same time, once it becomes intersubjective, it is beyond reason, not just reason alone. That is my take over kind of ethics that we need to develop to address this type of evolving, transforming technology which has many stakeholders, including life forms on Earth. So therefore, this is what I would like to tell you today. And I would like to thank you for giving me this opportunity to talk about this very important area of response that we humans have to develop.
Thank you, Father. After the valuable session by Dr. Victor Ferro, we are sure that all the participants have benefited a lot. This benefit has encouraged them to know more, and they have posted some important questions. So the first question, Father, is genetically modified organism a creation or just a evolution?
Genetically modified organisms are creation of human beings using genetic engineering. We rewrite the DNA, so we rewrite the code of life. We are able to, rewrite the code of life, and that's why we can have transgenic animals, plants, all type of even microbes. So we are having control over the destiny of all these life forms, and therefore we have to be very responsible in the use of this new technology.
Yes, Father. Now the second question, how do religions see gene editing?
Yeah, I come from religion. My position is, sometimes certain over-enthusiastic religious leaders may say, we are playing God. We are entering some area which is a dangerous area, and we are playing God. which I don't think is our response, should be our response, because God has given us an ability to be like co-creator or with God. And so we can bring all the potencies that are there in nature, in us, to the fullness, to the excellence. That is Aristotelian form that I was trying to embrace. That means potencies are there. Science also has potencies. We can technologically explore this, research this, and allow potencies to emerge to the fullness. But in this, we need guide, ethical guidance. We need virtue. We need a reason. We need also care. So these things cannot be done just blindly because somebody said, okay, this is the technology and we use. No, we need to be careful about it because we are going to transform not only our lives, but even the environment and animals. Everything will change.
This question answer, thank you, Father. This question answer session was really insightful, informative, and interactive. We are very grateful to Father Victor Ferro, who has given enlightening talk on ethics of gene editing. And I am quite sure our participants really benefited a lot. Thank you so much, Father, for taking time for this national webinar. Once again, thank you. Now, I hand over the proceedings to Father Job for concluding remarks. Father Job, over to Father Job. Father? Father, you are not audible, Father. Hello? Hello, Father, you are not audible.
Now I am audible, no? Now
yes,
I am
Father.
audible.
Yes, Father. Yes.
Yeah, there is please ask for Victor Ferravo, Dr. Victor Ferravo, to stop his sharing because he's already sharing, so I cannot get in to share.
Okay.
Please put it off, and so that I can start sharing. Please.
Yes.
Okay, thank you. Actually, we should have been seeing you earlier because of, anyway, doesn't matter. Just 1 minute. Give me 1 second to enter into my sharing this thing. Okay, I got in there. Okay. All right. Can you people see my slide?
Yes, Father, we can see. We can see your.
Very good. Okay, I I will put my concluding remarks in the context of some general ideas, which some general framework in which into which these 2 days experiences can be fitted. And first of all, I want to thank and congratulate all the paper presenters for the wonderful work, excellent work they have done. Yesterday, we had 2 presentations. Of course, the inaugural address of His Grace, the Archbishop of Nagpur, was very good. He had some very good ideas there. Afterwards, we had 2 presentations. Mine, of course, I have already spoken about. And Dr. Susan gave a very, very enlightening and detailed discussion of the latest and most powerful technology in genetic sciences, namely CRISPR. She gave a detailed analysis and discussion of the CRISPR with all its strong points and some of its weak points. That was very enlightening. Today we had 3 speakers, 3 eminent speakers. One, Dr. Hussain, and the other one, Dr. Srinivasu. And of course, the third one, Dr. Victor Ferrao. All these 3 sessions were also very enlightening. Dr. Hussain gave us how these discoveries are brought to practical life, brought to real life, particularly with regard to our food production, and very specially with regard to the hybrid varieties of maize. How these, this technological breakthrough has helped us to feed our stomach, feed our hungry people, to give them good food, nourishing food, enriched with the vitamins and other things. That kind of food, how it can be given. Then we had the session by Dr. Srinivasu. He gave us a detailed guided tour of this coronavirus menace. All of us wanted to know more about this coronavirus. It has been disturbing us, it has been troubling us, frightening us for almost a year. And he gave us a very, very detailed guided tour on this. And again, he gave us a roadmap of the science's fight against this coronavirus, the vaccine, the development of the vaccine, the roadmap of how scientists went about within a record time. Usually a virus vaccine requires about 6 years or more, some say even 10 years. Within 1 year, well, we have come up with a reasonably reliable vaccine. Tremendous success from the side of our scientists, hard work, ingenuity, all these things went into it. So he gave us a very beautiful picture of all these things. And so we had very enriching sessions, and of course, Dr. Susan and the principal of Elizabeth College and Dr. Susan's collaborators, all these people worked very hard to get these speakers and put these things together and make it a very successful 2-day webinar. So I want to express my appreciation, and at the same time, I want to congratulate them and encourage them to have more of these kind of events so that our people will be enlightened. Let me also mention that yesterday, today is of course we don't have the records, yesterday more than 500 people listened to us, more than 500 people, 100 plus from the Google Meet, and another 400 plus through the YouTube. And some of my friends called me up and saying, we were trying to get into the group, and we got the message that house full, no more people will be taken. So I told my friend, a professor, very eminent professor, please come a little early today and get in, otherwise you may not get your seat. You see, that is the type of response we got, and this is because of hard work put in by Dr. Susan and her team. And I want to congratulate all of you for the great work done. So let me give a few concluding remarks, which will be rather general and in some ways a personal thing. Please give me 5 minutes. I hope I will finish it in 5 minutes. Please give me 5 minutes. Okay, I'm sharing some of my thoughts. These are my personal thoughts. You can agree or disagree with me, no problem. And these are, open-ended claims or statements, and so they need to be modified and so on. Your critique and your response can help me to modify some of these things. Okay, let me begin. Science and religion, I believe, are God's gifts to humans. Science, in a way, is humans' grateful gift in return to God. Our world is an evolving, incomplete, and imperfect world. I believe that God has intentionally left our universe incomplete and imperfect so that humans can collaborate with God to complete and perfect it. Dr. Faravo mentioned this collaboration, human collaboration aspect in his presentation. Humans are invited to be collaborators with God in this noble mission of completing and perfecting the universe, our common home. Remember, we are We are, global beings. I would even say we are cosmic beings. So we can call our universe our common home. And a home means we should feel comfortable there. And also we have tremendous responsibility for the rest of the world, not only for ourselves, but also for others, and including our world around us. Okay. Humans collaborate with God in this mission mainly through science, by making use of the intelligence and
nutritional quality enrichment in maize through genetic avenues. Thank you, sir. We are grateful to Dr. T. Srinivasu, sir, for delivering a lecture on SARS-CoV-2 and its impact on religion. Thank you, sir. I would like to express sincere thanks to Dr. Victor Farrow for giving a talk on ethics of gene editing. Thank you, Father. Our sincere thanks to Father Patrick Limos, the administrator of our college, for his constant encouragement. Thank you, Father. I also thank our principal, Dr. K.T. Thomas, for his valuable suggestion and support and commitment. Thank you, sir. I would like to express wholeheartedly Father Praveen for the support and motivation. I also wish to express my gratitude to all the professors of Department of Computer Science for providing technical support. I thank Brother Daniel D'Souza, Mrunal, Ishan Shukla, and all students of SFS College who worked hard to make this event successful. Last but not the least, my heartfelt thanks to all the teachers of Physics Department for helping me and providing me the support to carry out various events of CDRIS. My special thanks to Sachin Guralwar and Madhu Gaur for the motivation they gave me. We thank all the distinguished participants for accepting our invitation. Finally, I leave you with this quote by Einstein: "Science without religion is lame, and religion without science is blind. Hence, we need both." Once again, thank you to one and all. I request all of you to stand for National Anthem.
जनगण मन अधिनायक जय हे पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे
To dear participants, the feedback link is available now. Please check WhatsApp group or YouTube or Google Meet platform. Certificates will be issued to those who submitted registration form and feedback of both the days. Thank you.
Thank